Hi, and welcome to Session 5, ACEMg becomes Soundbites. This Cards on the Table session covers translational medical research and how ACEMg negotiated the process, what worked, what didn't, what it cost, and what it achieved. I'll review the translational medical research process first. Feel free to skip ahead if you're familiar with it. Then I'll explain ACEMg's 16-year journey through it. If you watch the previous videos, you know how the story ends. ACEMg became known as Soundbites, clinically proven to preserve or improve hearing. I'll start with a summary of the process. The goal of translational medical research is to test the safety and efficacy of new drugs. In the U.S., the process is overseen by the Drug Division of the Food and Drug Administration. The European Medicines Agency does this for Europe. The process has two tests. Since drugs have side effects called adverse events, the first test answers the question, are the new drug's potential positive effects greater than its side effects? If a drug candidate passes that test, the second test answers the question, is the new drug statistically better than the current treatment? The FDA considers a new drug application if the clinical data says yes. If the application is approved, the new drug can be prescribed to patients by licensed medical professionals who monitor efficacy and side effects. Most people know translational research by the terms Phase 1, Phase 2, and especially Phase 3, the final human clinical trial phase. There's also a Phase 4. Phase 4 studies follow people after a treatment is approved to gather more data about long-term benefits or side effects that may not have surfaced during Phase 3 trials. The data help physicians, patients, and regulatory agencies. The process starts with Phase 1 safety trials conducted on small numbers of patients to determine dose range, aiming for the highest dose that doesn't produce unacceptable side effects. About 70% of new drug candidates survive Phase 1, which means about 30% fail, mainly because they're too toxic. By the way, all the numbers I'm sharing are aggregated approximations of historical data. Drug candidates that survive Phase 1 proceed to Phase 2 and Phase 3 random control trials, or RCT, to test whether the new drug is better than current treatments. In RCTs, people with the disease get either the new drug or a harmless placebo, the no treatment control. RCTs are double-blind, meaning neither the people conducting the trial nor the patients know who gets what. The drug and placebo are made to seem identical. The odds of getting the treatment are typically 50-50. The difference between treated patients and untreated patients is calculated as efficacy. Efficacy is weighed against side effects and is debated in the new drug application process. RCT trials take years. Phase 2 trials are conducted on small groups of patients, typically a few hundred. Historically, about 40% of drug candidates pass Phase 2, so less than 30% of new drugs go on to Phase 3. Phase 3 RCT studies are typically conducted on thousands of patients. Drug candidates that survive Phase 3 file an FDA new drug application. Review can take several years. In the end, fewer than 10% of new drug candidates survive translational research to become approved prescription drugs. Even then, success isn't a sure thing. The efficacy and side effects of the Alzheimer's drug Aduhelm, for example, were loudly protested during the approval process. The drug was ultimately approved, but it's being withdrawn from the market. Phase 4 studies are conducted after a treatment is on the market. These surveillance studies gather real-world data on the long-term benefits or side effects that may not have surfaced during Phase 3 trials. As we've seen, Phase 4 real-world evidence became significantly relevant to ACEMg research. Now let's take a close look at ACEMg's journey. The 2007 peer-reviewed lab findings confirmed that ACEMg insulated outer hair cells from noise damage and reduced hearing loss from noise by 75%. Clinical data was the next step. It's important to note that translational medical research wasn't a requirement for ACEMg for two reasons. First, hearing loss is a metabolic stress disorder, not a disease. ACEMg was designed to maintain or help improve the normal structure or function of the human body. Second, ACEMg isn't a drug. It's an inherently safe biomedicine. Toxicity isn't an issue. All the ingredients in ACEMg are identified as generally regarded as safe by the FDA and the U.S. Pharmacopeia. Presuming we could make the ACEMg formula into a product for RCT clinical studies, and demonstrate its effectiveness, ACEMg would be regulated as a dietary supplement by the Food Division of the FDA, available to the general public without a prescription. Nevertheless, we proceeded as if ACEMg were a drug because, coming from a medical school, we sought quantified effectiveness data and trusted the rigorous translational research process. Here's what we did. An investigational new drug application is required before starting translational research. The application describes the research plan and the drug in great detail. The ACEMg IND was 600 pages, compiled with the assistance of the University of Michigan Institute for Clinical and Health Research, or MICHR, submitted to the FDA in hard copy and PDF. IND regulations mandate pharmaceutical quality manufacturing, described in the IND section called Chemistry, Manufacturing, and Controls, or CMC. IND regulations require pharmaceutical quality manufacturing for the active and the placebo. Making ACEMg into a clinical study medicine turned out to be a 5-year trial and error process. We partnered with Patheon Pharmaceuticals. The first format was chewable mints, produced by Patheon Pharmaceuticals in Cincinnati, Ohio. Think Altoids for your ears. The mints inspired the Soundbites name, which stuck. But the mints failed as a medicinal product. Next, Patheon in France tried a sachet, a powder mixed into liquid. That format was abandoned because the active didn't mix very well, and it wasn't possible to formulate an identical placebo. Liquid was next, developed by Patheon in England. Think a 2-ounce energy drink for your ears. The formulation technology wasn't sufficiently advanced to achieve reasonable shelf life with guaranteed stability at room temperature. That technology now exists. We're developing soundbite shots for music professionals and fans who attend loud live events. Finally, Patheon Netherlands succeeded with softgel capsules and an identical placebo. The softgels ticked all the boxes: convenient, affordable, easy to swallow, identical placebo. The Soundbite softgels you can buy today are the same as used in the Phase 4 clinical study. The new softgel format required filing a second IND with the German Medical Regulatory Agency because our final RCT was being conducted at the Hanover Medical School, funded by the European grant. Next, I'll summarize the translational research studies themselves. The Swedish military study and the iPod study at the University of Florida were funded by the NIH. They were designed to test ACEMg to prevent acute short-term hearing loss from noise. But it's unethical to expose all subjects in a study to a known harm and only treat half of them, so noise exposure was controlled. Despite that, the Swedish military study returned some promising data. But the iPod study was a no test. There was no meaningful statistical difference in the data between the control and treated groups. The cochlear implant study at the Hanover Medical School in Germany also focused on addressing acute trauma. Surgical trauma associated with inserting electrodes into the cochleas of patients receiving cochlear implants. These patients were already functionally deaf. The study's aim was to assess the potential that pre-treatment with ACEMg could help the few remaining hair cells survive the trauma. It did. Patients receiving ACEMg recovered about 8 dB of outer hair cell auditory function. However, the trial suffered patient recruiting mismanagement, and the European government stopped the trial with about 17% of patients recruited. Unfortunately, 8 years of ACEMg RCT studies didn't produce sufficient clinical data. Meanwhile, ongoing lab research continued to yield promising data and new insights. A University of Michigan study demonstrated ACEMg mitigated SNHL from aminoglycoside antibiotics, a class of drugs used to treat gram-negative bacterial infections that upregulates inner ear free radicals as a side effect. Another University of Michigan study demonstrated that ACEMg mitigated SNHL from genetic mutations that account for more than half the cases of genetic hearing loss in children. Lab research at a university in Spain demonstrated ACEMg reduces apoptosis in inner ear cell mitochondria under oxidative stress, which helps explain the findings from the ACEMg real-world study. All was not lost. We'd gained 4 insights that helped define the research we needed to reach our goal to make ACEMg available to anyone who wanted it, backed by statistically significant clinical data. First, we learned to avoid short-term studies of acute noise exposure, mainly because it's medically unethical to purposefully expose humans to high levels of noise and only treat half of them. Second, persistent noise exposure is commonly understood to be an important contributor to progressive hearing loss. but the variety and conditions of exposure are vast. Instead of RCT, it's better to focus on uncontrolled long-term clinical surveillance studies comparing treated and untreated patients. Third, the ACEMg intervention must be administered in a convenient format, and the clinical study design for clinicians and patients must be simple and easy to follow. Fourth, expand the scope of research to include a broader range of hearing loss etiologies and include children, which would require a special dose form, which we make. My daughter gave me a change purse with Winston Churchill's quote that defines persistence. We pressed on. Everything we learned convinced us we needed to collect real-world evidence data in Phase 4 studies. These studies were possible because ACEMg was available in a convenient dose form without a prescription. You know from previous sessions that the ACEMg project moved from Amsterdam back to Ann Arbor, Michigan, and Soundbites started limited direct distribution in late 2019. About a year later, Chicago area audiologist Dr. Lori Halverson ordered Soundbites, urged her patients to use it, and started sharing de-identified data from their otoacoustic emissions examinations. The data demonstrated consistent objective improvement in inner ear function. Let me show you the data from one patient. This is the patient's initial OAE examination from both ears before starting Soundbites. Frequency responses go from low to high, left to right. Hearing sensitivity goes from low to high, bottom to top. This is data from the same patient at the 3-month time point. Hearing cells that didn't register as functional on initial OAE exams were coming back to life in subsequent exams, shown by the green check marks on the OAE test monitor. This isn't supposed to be possible, and it was happening consistently. The findings led to formalizing the study that yielded significant real-world clinical data. The next step is a scale-free clinical epidemiological study using OAE examinations to generate a larger data set for ongoing analysis and reporting to clinicians and public health agencies. For this step, we have a new motto inspired by a quote from William Gibson. The future is already here. It's just not evenly distributed. You can participate and stay updated at keephearing.org. I'd like to conclude with a chart that compares ACEMg discovery and development data with drug discovery and development data. Drug discovery and development typically takes 10 to 15 years. It took ACEMg more than twice as long, 36 years. Drug discovery and development typically cost between $1 and $2 billion. ACEMg total costs were about $30.6 million, about 1.5% to 3% of the cost for a new drug. Translational research for a new drug typically takes about 7 years and costs about $55 million. It took ACEMg a total of 16 years, more than twice as long as for a new drug. But it cost about 6 times less than drug translational research, about $8.6 million. Success odds for a new drug are slightly less than 10%. ACEMg is the first hearing preservation therapeutic we're aware of that attained the clinical validation milestone. Approved drugs receive a national drug code from the FDA Drug Division enabling prescriptions. ACEMg, Soundbites, is pre-approved by the FDA Food Division for sale to the general public without a prescription. Thank you for completing the course. You can contact me at hello@keephearing.org. Thanks again.